I'm James Weber. I'm part of the Methods Development Lab at the Broad, part of Broad Clinical Labs. We're out in force today because we're right across the street and you gave us food. And so feel free in the social time to find other members of the lab. I'll show them at the end and talk about other things that we're working on. So the Methods Development Lab, just as a short introduction, we are the birthplace of MOSSeq, which is now Connects from PacBio, but we're working on a lot of other things besides isoform sequencing. We're also interested in multiome sequencing, perturbation sequencing, all sorts of different methods, particularly in the sequencing space, particularly in long reads, because that's the best kind of reads. So my talk today is going to be about isoform sequencing, specifically single cell. So what does evaluating single cell platforms mean for isoform sequencing? Well, Revio and MOSSeq have really together made single cell isoform sequencing really exciting and powerful because the throughput is now there to get a lot of cells and a lot of isoforms per cell, and the full isoform rather than just the gene count. But all these methods that were built for gene counting in single cells, they were really optimized for that. And there's some artifacts that have been known in those methods that really maybe not a big problem if you were counting genes because you were still getting a good idea of the gene abundance. But if you're really interested in isoforms, particularly detecting interesting novel isoforms or things like that, these artifacts can really hurt you either because you're losing sequencing to reads that are not informative, or you're getting reads that are kind of misleading in that they are pointing you towards an isoform that is not real because it's a read that you captured through an incorrect mechanism. So in this study, we were looking at 3 different methods, 2 from 10x, which is kind of the gold standard in the field. They're 3' and 5'. And then this other method from Fluent Bio, which is a newer company actually in Watertown, just down the road, called PipSeq. And PipSeq is this particle templated emulsion method, which it's kind of wild that it works. Basically, you put beads and cells in a tube and shake them really hard, and that turns into a single cell suspension of beads co-encapsulated with cells, which, so I kind of cartoonified it as a vortexer, like the kind that you could just, stick your finger on in the lab when you were undergrad. And that's basically what it's doing, and it's really exciting because that is, as you can imagine, very fast and low cost and very, very scalable. You just get a bigger tube and a bigger vortexer, and you get a bigger experiment. So we really want to explore that method and also understand 10x 3' and 5' at the same time. And we did a short read and long read of PBMCs, and we also did a barnyard experiment of PipSeq that I'm not going to go into today. That was just to verify that the doublet rate is as low as we expected it to be in PipSeq. It is, and the ambient rate is not as high as you might fear for this method. It's actually quite comparable to 10x. Work? Yes. So this is the PBMC data. This is just the kind of normal short read single cell results. These are, if you're familiar with single cell, you're familiar with the knee plot, which is just on the x-axis the barcodes ranked from high to low, and on the y-axis the number of UMIs, the unique molecules per each barcode. In our experiment, we saw, we got more cells from PipSeq, and but we definitely got fewer UMIs per cell and fewer genes per cell. Some of that is a little bit experimentally specific, which I'm going to go into later, but this is also kind of what you would expect from the 10x is this microfluidic platform that has an inherent limit on how many cells you can put in there before you get unacceptable levels of doublets. So we have about 2,000 cells, which is kind of what they recommend. A lot of people go higher, of course, but you're getting more doublets that way. And we have about 7,000 cells from the PipSeq experiment. I should say all of this data is subsampled to 100,000 reads per cell. So this is the same sequencing depth per cell in these experiments. So, we see that our UMIs per cell and genes per cell are a little lower, but we can also look at the kind of biology of this sample. So these are PBMCs. They're not, the most exciting new biological system, but they're very well studied and well understood, and we see all of the kind of expected cell types with the expected markers in all three of these. Technologies. The clusterings are slightly different here, and part of that is because, for instance, 5', which is really almost was developed for immune cell profiling, does differentiate some types of T cells that you don't differentiate in the 3' profiling, which PIP-seq is also a 3' profiling technology. There's also, just differences in the number of cells that were captured, and there's also differences in ambient rate. In our PIP-seq experiment, we do see higher ambient in this experiment, and again, I'm going to go into this later. So the next slide, we were really interested in long-read sequencing, and specifically the artifacts that we see in these libraries. And so what we did here is we took all of the cDNA from those 3 different libraries, and we did basically what we call monomer long-read sequencing, but basically isoseq. So each, one cDNA per read on a sequel 2E, and we used some computational tools that we developed to basically annotate the adapter sequences on the 5' and 3' adapters and the poly A tails to say what kind of read, was this a properly formed read? Does it have the 3' adapter, poly A tail, cDNA, and a 5' adapter? Or does it broadly have one of these 2 artifact types? So there's TSO, which is the template switching oligo. You can imagine, so basically you prime from the 3' end, you add a template switching oligo, and then somehow another template switching oligo sneaks into the other side, and you end up with this double-ended read where it's 5' on both ends. The adapter is the 5' on both ends. And you can imagine that if you're trying to make an array out of this is going to cause a big problem. It's going to make your array kind of a palindrome, and it's going to mess up your array formation. And similarly, you can have an RT artifact where you have the poly A, you do reverse transcription, and there's a poly T somewhere in your isoform that another RT adapter just comes in and sits on that one, and you get this other double-ended cDNA. So we knew previously from that 10x had this really high rate of TSO formation, which is, so if you've done connects, there's a TSO purification step that you would just have to do to get a good array out of that technology. And so what was exciting to us is that in this PIP-seq experiment, the artifact formation for TSO, and RT was a lot lower. In fact, so low that we could skip that step and still get good arrays. There is this other category of artifacts, and so I guess I'll say now, so the main category of artifacts that we saw in PIP-seq was what we call all poly A. So it's basically just a poly A read. There's no kind of mappable cDNA. And this is not, this is a kind of specific to our sample, and we're going to talk about that in a little bit. But basically, this is degraded RNAs that are, so you're really just sequencing the poly A tail and not getting anything out of it, and that's because of degradation during the sample processing. So we did take the PIP-seq, so one of the headlines that I want to take away is that this PIP-seq technology is compatible with MOS-seq. This is something that hopefully will be coming soon, kind of more broadly available. So we did basically 2 SMART cells worth of sequencing, which was about 6.8 million hi-fi reads, translated to over 100 million segmented reads from MOS-seq. And I should say that this was quite a while ago. Our Revio has gotten much better, and we've gotten much better at getting data out of it since then. So I think this is maybe literally half as much data as we would get today if we did this experiment again. And these 2 figures are just showing kind of QC plots from the MOS-seq, showing you that almost every read that we got was a full 16x array. All of the ligation pairs are almost literally everything was paired up as you'd expect, which is just a good sign that the segmentation worked properly. So now I'm going to get into kind of the sad part of our data. So as I mentioned, there is this RNA degradation artifact, and this is particularly noticeable in monocytes that are expressing RNases really highly. And so if we could see this in our short read data. That formatting is unfortunate. We could see this in our short read data that these monocytes had a lower number of UMIs per cell, but it was still kind of above our thresholds to call them as cells. But in our long read data, that's 10 to 100 UMIs per cell. You would normally throw that out of your experiment as an ambient droplet. We only knew that was a real cell because we had the short read data that we had called it. And when we go to our, back to our pre-processing and look at reads that we had thrown out previously, because they were too short in the long read context, we can actually get those cells back up to a reasonable number, but those reads are not really mappable. They're these poly A reads that I was discussing before, so you can't kind of get good counts out of them. And certainly in the context of isoform sequencing, these short reads are not going to help you call any isoforms or count genes or count anything interesting. So, and another thing that's really interesting about this is that this was definitely differential across different monocyte populations. So it's something to do with specific RNases and something about the PIP-seq process that are really stressing some of these cells and causing them to rapidly degrade RNA. So how do we fix this problem? We literally fix the problem. So this is data from Gami Eliani, who's a member of the lab. He did a panel of different fixation methods and tested them all out in one hashtagged PIP-seq experiment. So he mixed them all together, and then we had made MOS arrays and did long-read sequencing. You can see this really nice effect from in only the CD14, so this is specifically in the CD14 monocytes. The other cells that are broadly unaffected kind of just look the same across the different conditions. But these methanol fixation methods really rescue the number of UMIs per cell. They really decrease the number of poly A reads that we saw. And then if we look at the read length, they really rescue the read length in those cells as well. So that it really seems to be preventing this degradation from happening before it gets into the PIP-seq method. PFA, which is another fixation method that you might try, because it's cross-linking two nucleotides also, really is not the way to go here. You want to avoid doing that. So we were really happy with this. We're hoping to kind of explore this fixation strategy more to make PIP-seq like a much more viable method. But next I'm going to go into kind of isoform identification and quantification, because that's what you would do with these technologies. We used isoquant here, which is, there's a lot of different tools out there for isoform identification and quantification. This is one of the ones that we found has been performing well. But I should say I'm not showing this to brag that we found 50,000 novel genes in human genomes, because I think we all agree that's probably not true. I'm actually showing this to highlight how difficult this problem is, especially in the context of single cell, where there's all these artifacts that are really confusing to these tools. And so, these results are actually quite good compared to some other tools that you can run on these data. But single cell, because the counts are very low and the data has a bunch of different artifacts that have not been fully explored and understood, it can be very difficult to run this isoform identification. And this is all the same sample, right? And just run on 3 different platforms, you can get quite different results in terms of, so the known genes and known transcripts are quite similar, but the novel ones, because of different artifact profiles, can be quite different. Nonetheless, we can look at least the known isoforms and try to do some differential isoform analysis. So here's an example of Fc gamma receptor 3A, which is CD16. It's a cluster differentiation. We can see that across these 3 different clusters where it's expressed, we have this really nice separation of isoforms, where 205 is really in the monocytes, 201 is in these 2 different T-cell clusters. But this gets me to another lesson for my talk, which is the ABC rule, which is always be checking your read support. So what was really happening here, if you look at the read support, so there's 2 things to kind of, is this a laser pointer? I don't know. There is a slight difference in the 3' end of these 2 isoforms, and you can look at the read coverage. There's kind of a similar dip in all 3 of them, but there's also a difference in the 5' end, and this exon is missing, and that's what led, in terms of read assignment, to the CD16 monocytes only having that 1 isoform. But if you look on the 3' end, it looks a lot more similar. And so what I think is really happening here is that there was more degradation in the CD16 monocytes, which we are not surprised about, and due to read assignment and quantification, it assigned all these genes preferentially to 1 isoform, and there's a lot of ambiguous reads that it didn't assign to anything. But when we look at the coverage, overall, it looks like a lot more kind of equal. And so this is just another cautionary tale to say that really look at the data, this problem is not solved, and we really need, we're working on better tools and understanding all of this better. I do have a happier example. So CKLF is this chemokine-like factor. It has 4 isoforms, 2 of which are membrane proteins, 2 of which are secreted. We have really beautiful kind of end-to-end coverage on this gene, where you can see all 4 isoforms, the full length. Exon 3 is not included, exon 2 is not included, both 2 and 3 are not included. There's differential usage across the 4 cell types, and this is really an example of where MAS-seq and Revio come together to really give us something new, because this is not a super abundant gene, you really need the depth and you need the single cell resolution to differentiate between different cell types, and you need the depth to be able to see this confidently across, to see the differential usage across these 4 different isoforms. So with that, I'm going to summarize everything I've said. So one of the things we wanted to share and to show is that PIP-seq is this new platform that's really promising. It is compatible with MAS-seq and with the Revio, and it really opens up a lot of new possibilities for single-cell isoform sequencing. In particular, I personally have not experienced this, but I'm told that this TSO purification in Kinnex is really a pain. So thank you to Dan and Bartlett and everyone else in the lab who actually does that. But in PIP-seq, you don't have to do it because the artifact rate is sufficiently low that you can still get good arrays. We're really interested in using long-read sequencing also just to better understand these platforms, because the artifacts that are created during library preparation are actually critically important, especially for isoform identification. And even just in the context of short-read sequencing, they can also be really informative to tell us how those really high counts that we're getting out of these new technologies might be biased in different ways. And really, my final message is just that we're just at the very beginning of single-cell isoform sequencing. It's really exciting time because the sequencers are getting really high throughput, the methods are getting better, and we're starting to understand all of the different kind of characteristics of this data. And it's just going to get more interesting, and we'll be able to find more and more interesting things that hopefully will have more clinical applications than I showed you today. So with that, I would just want to acknowledge everyone in the lab, and also Victoria Nolia, who helped us with this artifact detection. I'm really sorry about the spacing on the fonts here. It looked good on my machine. But yeah, and I'm happy to take any questions.
Thanks, Dr. Weber. The mics are open.